All right, so this course is about applying laws of physics to the life sciences. Okay. In nature, what does physics laws apply to? Everything. You can't build anything without obeying the physics laws of physics. Okay, so we start off with, in nature there's so many different kinds of life forms, but every one of them has to obey laws of physics. Okay, so I'll give you. So here's a. We'll start off with a little example. So, let's say here's a blood vessel. You want to control the flow rate of blood. Okay, and this is pressure 2, pressure 1. Again, don't worry about the formulas in none of the exams. You don't have to solve formulas, but you just need to get a sense for it. Okay. And again, I'm just putting formulas just to illustrate points. You don't have to. Again. Okay, this is just a formula. Okay. I'm trying to make a point here. Okay, so here's a blood vessel. Here's blood flowing. Okay, so let's say you go for a run or you do some intense workout. What is it that you want for the blood flow to your muscles? Increase. Okay, your blood flow can increase by a factor of 5. What that means is, if I'm just making up numbers, if a gallon a minute are flowing during intense exercises, 5 gallons a minute will flow. So you want to increase this flow rate. How do you increase this flow rate? You want to increase this number, right? That's the radius of the blood vessel. This is the pressure difference. So, if you want to increase the flow rate by a factor of 5, what you could do is you could increase the pressure difference by a factor of 5. You can make your heart pump with the force 5 times greater. That's a stupid body like mine that'll do it. How else can you increase the flow rate by a factor of 5?
By increasing the size of the blood vessel?
Thank you. That's a smart body. All you have to do is increase by about 15%, and you can increase it gets to the power of 4. And that increases the blood flow rate by a factor of 5. That's what a conditioned athlete does. When you're training your body to increase the size of the blood vessel and not having to make your heart work as hard. Okay? So this is the kind of stuff we want to learn. Okay? Do you guys understand? Okay, so that'll be the point of the course. We are going to apply physics principles to life sciences. Yeah. Your bodies are very smart. All life is very smart. If you want the best solution to any problem, you go to nature. Nature has the best solutions to all the problems. Why does nature have the best solutions to all the problems? How come nature has the best solutions? Yep. I'm sorry? Well, it's had millions of years to perfect every solution. And if it doesn't have a good solution, it just kills the animal. Okay? All right. So, like I said, biology has some of the best solutions. Okay. So, we're going to apply physics principles to the life sciences. Okay? So, how physics sheds lights on biological processes. That's the goal of the course. Okay? So, we'll start off with with an MRI machine. Here's the image of head, okay? A slice of an image of the head, okay? What does MRI stand for? It says magnetic resonance imaging, okay? How many of you have had an MRI here? Okay. All right. So, I mean, everybody's heard of MRIs, and many of you have had an MRI. Okay, so let's say here's my head. An MRI can take a picture of that spot inside my head, and then it can take a picture of that spot, that spot, that spot, and then it puts together a three-dimensional image. What this is a slice of that image. Yep. The resolution, the smallest picture it can take off is roughly a small grain of sand. Okay, 0.5 millimeter by 0.5 millimeter by 0.5 millimeter. Okay. Right. Now, what we want to do is understand how does the MRI machine take a picture of that little grain of sand inside your head. Okay, so we'll try and understand that. Okay. You guys know there are several levels of MRIs. Okay. So, and so the MRI machine, there's a magnetic field in this room, the Earth's magnetic field. And in the MRI machine, there's a very strong magnetic field in there. That's about 10,000 times stronger than in this room. Okay. So the magnetic field in this room, these are just units, is about 0.5 times 10 to the minus 4 Tesla. That's roughly, okay, we'll say that's 1/10,000th of a Tesla. The MRI machine in this, the magnetic field in this MRI machine is about 1.5 Tesla to 3 Teslas. Okay, so about 10 to 20,000 times stronger. Okay. Let's say you flip on the MRI machine, and if you're not careful, this is what can happen. So it's a very strong magnetic field. Okay. The stronger the magnetic field, the finer the image, the better resolution you can get, and the more expensive the MRI. Okay. So so there are MRI machines that use 7 to 10 Teslas, okay, for specialized application in neuroimaging. Okay. So again, the stronger the field, the sharper the image. Okay. All right, so the MRI machines that we use is about 1 to 3 Teslas. Okay. So stronger fields provide higher signal to noise ratio, and so you can use that to get better resolution. Okay. All right. So MRI scanners can identify tissue types of a cube that is 0.5 millimeters on each side. Okay, so that's roughly, like I said, a small grain of sand. They can take a picture of a small grain of sand, and each grain they take a picture, then you take the picture of the next grain, and then put together a 3D image, and then you can see any slice you want. Okay, so if you want to see a slice like that, like that, like that, okay, once you have the 3D image, you can see any slice, okay, and diagnose whatever problem it is. All right, so now I'll briefly tell you how the MRI machine works, okay? Okay, you guys know the hydrogen atom. Okay, so here's the hydrogen atom. Okay, so what is this?
Proton.
That's the proton, there's the electron, okay? So that's in the nucleus. This fella, proton, is 2,000 times heavier than that. So all of the mass of the hydrogen atom is essentially in the proton. I'm sorry?
It's a lot bigger than like a normal one, right?
What do you mean, lot bigger?
Like if it's like size difference or something.
Yeah, okay, so what our friend here is saying is, okay, the atom is very tiny. Have you ever seen an atom? No, you'll never see an atom. Okay. It's tiny. This fella here can confirm that. Okay. The nucleus is tiny. So if, okay, I'll give you the actual numbers, but so 10 to the minus 10 meters is the size of the atom, and the size of the nucleus is 10 to the minus 15 meters. Okay, now let me give you more of a easier example to understand. If the atom were as big as a football stadium, the nucleus would be the size of a marble in the stadium. Okay? Now the MRI machine uses this fellow to get the image, the proton. Okay, so now again, with all these stories, you have to be a little patient, and they're more than 10-second stories, so you have to follow me along. Okay, the proton is spinning. It's a charged particle. It's spinning, and what the spin does is it acts like a magnet. Okay, so all these protons are little magnets. Okay. Does your body have these hydrogen atoms?
Yes.
Where does your body have a hydrogen atom?
Within the water that's inside us.
It's in the water inside of you. Okay, so here's the water molecule. I like to call it a Mickey Mouse molecule. Okay, H2O. Okay, here are the two hydrogen atoms, and here's the oxygen atom. And so your body is full of hydrogen atoms. Okay? And in the hydrogen atom, these protons are there, they're spinning, they're acting like little magnets, and so your body is full of these little magnets. Okay? And we're going to use that to get the MRI image. Okay? All right, so in this MRI machine, they have a very strong magnetic field. Like I said, that magnetic field is 20,000 times stronger than the field in this room. A very strong magnetic field. Okay, so the magnetic fields are denoted by B. Okay, the letter B. If the proton, that magnet, lines up with the magnetic field, then its energy is low. If the proton points in the opposite direction, it has a high energy. Okay. Okay, so now we're getting to the punchline. Okay. So what is it that we want to do? We want to take a picture of this little spot inside your head. Okay. All right. So here's the proton that's lined up like that, parallel to the magnetic field. It has low energy. If you can somehow make it line up opposite, you can flip it, then it'll go into high energy. Okay? So what you do is you send a pulse of light, a radio frequency light, that will flip the proton from here to there. So now the proton has gotten excited. Okay? So what you did was you sent a pulse of light to that spot. That proton got excited. Okay? And now, soon, very fast, within a billionth of a second, that proton will emit that photon again, that radio pulse. And you use that, you detect this to image that point. You're seeing the light from that point. Did you guys understand that? Okay.
You said it sent a light, what was that?
So you sent this photon, okay? And I hope you're with me, because we're getting to the punchline, and this is very cool. Okay, we're figuring out how you're going to take an image of a point inside your head. Okay, all right. So you sent this photon. This proton that was there absorbed it and then emitted the light, and you detected it, and that's what you used to make an image. Now here's my question to you. You sent this photon. Why didn't all these guys absorb that photon? How are you going to make sure that only that guy absorbs the photon? Did you guys understand that? That's the crux of the matter.
Would be the frequency.
I'm sorry?
Like the frequency?
Okay, so you understand the question, right?
Yeah.
Okay, so here's like so okay, so when you send this photon, the photon has to have an energy exactly equal to the energy difference between these two levels. Okay, only those guys can absorb that photon. And this energy difference, delta E, again, don't worry about any of the equations, depends on the magnetic field strength. Okay, so if you want this guy to absorb that photon, you have to make sure that at that point precisely the magnetic field is correct. Everywhere else the magnetic field is different, and none of those hydrogen atoms can absorb the photon. Did you guys understand that? That's the simple trick. Okay, so here we go. So what you do is, so here's your head, okay? There's a strong magnetic field in there. You already saw that. And then on top of that strong magnetic field, you apply gradient fields so that the magnetic field at every point is different. And at the point that you're taking a picture, it precisely matches the radiation you're sending it. And only that point will absorb the radiation and emit it back, and you can take a picture. Okay? And then if you want to take the picture of the next spot, you change the magnetic field, go to the next spot, make the magnetic field appropriate there, and take a picture. So when they take an MRI machine, and when you're taking an MRI of whatever body part they're taking, they stick you in the machine for 20 minutes. And why are they doing that? Because they got to take millions of pictures. Okay? And the computer is putting it all together. Okay? You guys get a sense for the MRI machine? All right. All right. All right, so I've already explained this to you, so you guys can read some of that stuff. Okay. What does a dialysis machine do? Yep. I'm sorry? It filters and cleans the blood. Why do you have to do that? That's right. Okay. So in place when your kidneys you're born with lifetime warranties on all your organs, but if sometimes some of your organs fail, then, right, so you have to get dialysis, filter your blood. Okay, let's figure out how this is done. Okay, so you have to understand something called diffusion, and this is a very important concept. Every transport, anything that's transported in your body, the distance of a cell, the process involved is diffusion. Okay, so let me show you a little thing for diffusion. Yeah. All right, we'll just do it like this. Okay, so so imagine the following. So this is blood with toxins. Okay? And this is pure water on this side. Okay, so this is urea and creatine and all that kind of stuff. Okay. And what we're going to do is we are going to make this a semi-permeable membrane. What does a semi-permeable membrane mean? Yep. Very good. Thank you, my friend. So like the name says, semi-permeable. It allows certain things. It won't allow everything. Okay? So the membrane has holes, and it won't let through large stuff. It'll let through small stuff. Okay? So and you see, via random processes, these molecules are diffusing through there, to there. Okay? Now, for these molecules to go through from here to there, what needs to happen is the concentration of those molecules needs to be higher here and lower on this side, and that go through there like that. Okay? And when does the diffusion stop? When the concentration equilibrates. Molecules are still being exchanged, but there's no net exchange of molecules. Okay? And that's diffusion. And that is the process that's used in your body when molecules have to be moved small distances. When oxygen has to flow across the cell, it's through diffusion. Okay? When oxygen goes from capillaries to cell, it's through diffusion. When neurotransmitters are transmitted across the gap between two neurons, it's diffusion. Okay? But this process works only for small distances. If you have to transport molecules long distances, for instance, if you want to transport molecules from your lungs, oxygen from your lungs to your legs, how do you do that? I'm sorry? No, then you have to go by plumbing. Then you have to go by plumbing. You have to have blood vessels. Blood has to carry the oxygen from the lungs to the skeletons. To the muscles in your legs. And the capillaries, you have to bring the blood close to the cells, and then diffusion can happen. Okay? Do you guys understand that? Okay, so diffusion works only small distances. Soon as you become large, then you have to have a circulatory system to transport stuff closer to the destination. Okay, so everybody understand diffusion? All right, let me redo that again. Okay, this time. So this is bad stuff. Semi-permeable membrane. The higher the concentration difference, the faster the motion of the molecules. Okay, and I'll give you numbers on how long it takes in a second. Okay, but I want you to first understand this. We'll make this semi-permeable membrane, and it will go through there. Okay, so we understand this. So now let's go to the dialysis machine. Okay, so a dialysis machine is an artificial kidney. It has waste products diffused through a membrane from blood into a dialysis liquid. Okay, so again, so now you guys should be experts at dialysis. So here's what's happening. Okay, so here's your blood vessel with toxins. Oops. And here's the dialysis fluid, and these are semi-permeable membranes. All right, so here's the toxin concentration is higher. Here's a semi-permeable membrane. What is the toxin concentration in the dialysis fluid? 0 or less, okay? And as the blood flows, all these toxins diffuse into the dialysis fluid, okay? And then you got pure blood here, okay? That's the idea. So you're using diffusion to remove toxins from your blood in a dialysis machine, okay? All you have to do is, as this thing what do you have to do as the toxin flow in here? You just have to flush it out constantly so you can keep the diffusion process going. Okay? All right, so in hemodialysis, many waste substances move from the patient's blood into the dialysis fluid. It's called a dialysate, primarily by diffusion across a semi-permeable membrane. Okay? So you're using diffusion. Molecules such as urea, creatine, potassium, and phosphates are relatively high concentration in blood. Okay? The fresh dialysate, okay, where there's no urea and all that, okay, that's in the dialysis machine, and all this toxin diffuse across the membrane. The fresh dialysate is formulated to have lower controlled concentration of these substances. Okay? And these molecules diffuse across the membrane. All living organisms have to obey everything in nature has to obey the laws of physics. Okay? Energy such as laws such as energy conservation. What does energy conservation mean?
Energy,
I'm sorry?
saving energy.
Here what we mean is that you cannot create or destroy energy. Okay? So if you want to do work, you need energy. And the units of energy are joules. If you do 100 joules of work, you have to have 100 joules of energy. Okay? So everything we do in life, so for instance, okay, I can increase the energy of this guy by doing work on it. Okay? When I lift this object up, so let's say I increase the energy of this guy by 50 joules. What happened? I spent 50 joules doing work, and I transferred the energy from here to there. Okay. So everything we do in life amounts to moving energy from one object to another. That's all. You're not creating energy or destroying energy. Okay. So every life form has to obey laws of physics. Okay. So energy conservation will be a principle, and okay. So fluid flow, you saw an example of the law that governs fluid flow and all. So we will be using that. We see that biological systems have evolved spectacular diversity within these constraints. Okay, so that's a powerful statement. Life has evolved so many different forms, every one of them obeying the laws of physics. Okay. Here's a image of a scanning tunneling microscope image of tiny objects. So physics provide tools enabling image and measure cells and organisms ranging from viruses to humans. Okay. The scanning electron microscope image shows salmonella again, invading. Again. So we want you to understand living systems and biomedical tools. Okay. So how many of you have heard of a scanning tunneling electron microscope? You've heard of them. Have you seen one?
Yes, I've actually gotten to use one.
Oh, really?
Yes.
Where?
It was at a college preparatory school that we were visiting, and they were performing some science experiments with the scanning electron microscopes.
That's pretty.
And I got to see a leaf under one of them. It was actually wonderful.
All right. Anybody else? Tell me your story. What?
It was just in class.
You saw it in a class?
No, like,
Okay. You heard about it.
yeah.
Okay.
Class.
You guys want to know how that thing works? Yes? All right. So this thing is also called a scanning. Here we call it a scanning electron microscope, but it's also called a scanning tunneling microscope. Okay? And you'll understand the name in a second. Okay, so let's say we want to take an image of an object that's here, placed on the table. Okay, so let's say we want to take an image of this object. Okay. So what you have is you'll have an electrode like this. Okay. And what you'll do is let's say you will connect 25,000 volts across it. Yep. And if let's say there's a bump here on this object, what happens is, so this is the negative terminal of the battery, this is the positive terminal, and if you connect a high voltage, what will happen is electrons will tunnel through this gap. And that's why the name scanning tunneling microscope. All right. So the smaller the gap, what will happen to the electrons? The number of electrons tunneling will go up or down? Up. Right? So when this probe is on top of here, there's a higher current, and when the probe is here, the current is low. So current is denoted by I, high, current low. So if you're taking the image of this object, all you're doing is you're scanning this probe, the electrode, by maintaining its level across that surface and just measuring the current. What does a high current mean? I'm sorry? A high current just means there's a bump there. A low current means
There's nothing there.
it's a flat spot. Okay? So this image that you see, it's just proxy for current. This was a high current, and that was a low current, and so on. Okay? All this color was added later. Okay? So all you're doing by using the scanning tunneling microscope is you're just measuring currents at different points. Yep.
So does it also create a magnetic field?
No, you're just measuring currents. Okay.
Well, sometimes you could use magnetic fields to move the electrons for the scanning part. Measure where else it lands.
No, you're scanning the electrode. You don't do that. Okay. All right. So anyway, so that's the scanning tunneling microscope. Okay. So you can see tiny objects. You can see objects as big as 10 or 15 atoms. Okay. So physics provides those tools. Biological processes are physical processes. Okay. They help you understand a great deal about biology. You guys know what are the size of capillaries? Okay. Capillaries are, the size of capillaries is such that only a single red blood cell can flow through it, one at a time. Do you know why that might be? That's when the friction is the least. If this was slightly wider and maybe 1.5 could fit in, then there would be higher friction. And if there's higher friction, what's the problem?
Blood clot?
No, your heart would have to work harder.
High blood pressure.
Okay? So everything is designed for good reason. Okay? All right. So the circulatory system from heart to capillaries is governed by fluid dynamics. Okay? Neurons signal each other via electric pulses that travel along axons. So there's the axon. Okay? How many neurons do you have? Do you guys know what is the most powerful computer in the world?
Brain.
Yep. There's one attached to every shoulder. It's 90 billion interconnected neurons. Okay? The more connections you have, the better you understand stuff. And when you're learning stuff, you're making new connections. Okay? Okay. Have you guys heard of fluorescent proteins? They have different colors. So this is a green fluorescent protein. The person who invented that, won the Nobel Prize. Anyway, now they're different colors. So here's stuff inside a cell, and they're taking pictures. How do you see through a cell? Okay, or how do you see through anything? Okay, they can put these green fluorescent protein inside your skull, inside your brain, and take pictures. How do you do that? Okay. I'm sorry?
MRI machine?
Well, yeah, that's one technique. Here's a
X-ray. Oh.
no, not X-rays. Okay, so it sort of has to be transparent, so you can't do that. Okay. So here's the idea. So here's a cell, and you want to take a picture of this organelle. Okay. What you do is you take a molecule and attach a fluorescent part to it. Okay. So so here's even though I painted it red, it's a green fluorescent protein molecule. Now what you do is you shine UV light on it. UV light, and again I'm simplifying stuff. Okay. This molecule absorbs the UV light and emits green light. Okay, now the light being emitted is different color than what you're shining. Okay, so you're swamping this stuff with this light, but what you're detecting is a different light, so you can take a picture of that. Okay, so that's how you take a picture of these guys. Okay. You want to take a picture of this organelle, what do? You just attach a green fluorescent protein to that organelle. And that's how this picture was taken. Okay, so you attach these fluorescent molecules. Okay? Nowadays there are green, red, blue. ChatGPT. Okay, so find out all the different. Okay. Do you guys understand how that picture is taken?
Yes, sir.
All right. So now we'll talk about fluorescence. Okay, the process itself. Okay. All right. By the way, so physics will tell you what are the limits of athletic performance. Okay, so here's fluorescence. Okay. A photon is a light particle. The energy of a light particle is h, the Planck's constant, times the frequency of the light. Okay. UV light has higher energy. UV light has higher frequency and it has higher energy. Visible light has lower frequency than UV light and it's less energy. Infrared light is less energy than UV light, than visible light, and it's got less energy. Okay, so let me show you the electromagnetic spectrum. Right, so we have sensors to detect electromagnetic waves. Your eyes can only sense those wavelengths. Infrared light is longer wavelengths. UV light is shorter wavelength, higher frequency, and so on. Okay, so we use various kinds of waves for different things. So you use X-rays to take pictures of your bones. ultraviolet light, visible light, infrared light, and so on. Okay? So again, if you go in this direction, energy increases. Okay? So they're all electromagnetic waves, and they're called electromagnetic waves because they have both. So that's a light beam traveling in that direction, and it has both electric fields and magnetic fields. Okay, in it. That's why it's called an electromagnetic wave. All right. So coming back to fluorescence. Okay. So remember, we are taking a picture of that inside the cell. What we're going to do is shine UV light on that molecule, the fluorescent molecule. Okay. That molecule gets excited. Okay. So that you shine UV light, it has high energy, that molecule gets excited. And then it loses some of its energy via vibrational modes through heat. Okay? So it loses some of its energy. And then, so that molecule is complex and it has many energy levels. Okay? You excited it, and then it de-excites by emitting this green light. So the importance was, when you shine a UV lamp, you're swamping it with UV light, but what it's emitting back is a different color. And you can detect that. You train your camera to see just that, and you can take a picture of that. Okay? So you apply filters on your camera, everything else is filtered, only this light is coming through.
Like how we need to have IR filters because most camera sensors are more sensitive to IR than
Right.
visible light.
Right. So you shine UV light on that and have filters and take just a green picture. Okay, so that's how that image was taken. Okay. And this way you can study various molecules in different parts of the cell. Okay. All right, so in fluorescence, a molecule absorbs a photon, ultraviolet or visible light. This energy boosts an electron from lower energy ground state to a higher excited state. The excited electron loses some of its energy via vibrations. That's, you can think of it as heat, going to a lower level. Okay. This energy loss is not emitted in light, but is lost as heat. Then the electron drops back to the ground state within a billionth of a second. So all this stuff is happening very fast. Yep. You're shining UV light, and within a billionth of a second, you're getting this lower energy green light. Okay? Okay. So here's some of the places you can use it. Okay, so in forensics, detection of bodily fluids. So there might be small amounts of bodily fluid. You shine UV light, and the fluid, some specific fluids are fluorescent, and they send a light of a very particular frequency, and you detect that. Okay. So it's a very fast process, common in organic molecules. Many organic molecules are fluorescent. Okay, so you can shine high energy radiation on it, and they will fluoresce. Okay. So you can use this technique to identify minerals. Fluorescent ink is used in currency and documents. Okay. So there's fluorescent ink, and that molecule it won't degrade, and a fake currency won't have fluorescent molecules. So if you have a real currency, you just shine UV light on it, and if it fluoresces, then it's a good currency. Okay. Okay, so redwood trees can be 100 meters tall, the size of a football stadium. Okay. And, the question is, how does water rise to the leaves of those trees? And, physics explains how water will rise through the xylem. Okay. Now we'll understand this device. What does this device do?
Measure blood oxygen.
I'm sorry?
Measure blood oxygen.
Right. So it'll measure What carries oxygen?
The red blood cell.
Hemoglobin binds to oxygen. Okay. So when they say your blood oxygen level is 95%, what does that mean? I'm sorry? 95% is low? Okay. All right. Anyway, what does that number mean, 95%? Huh? Okay. No, you got to understand stuff. Okay, so when they say blood oxygen level is 95%, here's what it means. Okay, if you have 100 hemoglobin molecules, what do you want them to do? You want every one of them to carry oxygen. If every one of them is not carrying oxygen, then some guy is not carrying their weight. 95% means 95% of the hemoglobin molecules are carrying oxygen. Okay? 100% means 100% of them. And you definitely don't want to be below 90%, right? It's something like that. Okay, so this guy measures how many of the hemoglobin molecules are carrying oxygen.
96.
I'm sorry?
96.
Okay, well, I mean, that's just a number. We want to know how it works.
Oh.
Okay? So here's and again, when you understand this, when you guys are out in the field, you can invent stuff, okay? Invent an instrument and become billionaires, okay? And come back and give me some of that money. All right, so here's how this thing works. All right, so this is, so a pulse oximeter measures the percentage of hemoglobin in your blood that is carrying oxygen, okay? So it can also measure pulse rate. Okay, so the device uses two different wavelengths. So here's the principle. Red light. Okay. All right. Red light whose wavelength is 660 nanometers. So again. Yes, the wavelength is 660 nanometers. So here's the visible spectrum. That narrow band is the visible spectrum, and that's been expanded here. So this is longer wavelength, that's shorter wavelength. This has around 660 nanometers. That's the wavelength. Okay. So red light is absorbed more by deoxygenated hemoglobin. Okay, so hemoglobin without oxygen absorbs red light. Infrared light, 940 nanometers, is absorbed more by oxygenated hemoglobin. Oxygen that's bound to hemoglobin, that gets absorbed by infrared light. Okay, so here's what we do. So again, there's the visible light. Here's infrared light. Longer wavelengths get absorbed by oxygenated hemoglobin, oxygenated blood. Okay, so here's what we'll do. Here's your finger. Here's blood flowing, and what we'll do is we'll shine an red light and an infrared light. Okay, so if your blood is, I should use different colors here. So here's blood flowing. Okay, so if your blood is oxygenated, if all the hemoglobins are carrying oxygen, what happens to this light? Infrared gets absorbed, and this intensity will be low. if if your blood is carrying no oxygen, this light gets absorbed and this will be low. Did you guys understand that?
So we're measuring the difference between
That's
the
all.
light and the IR.
Okay, yeah. So you're shining infrared light and red light from the bottom, and at the top you're measuring it. And in infrared light is low, that means your blood is oxygenated. And if the red light is low, that means it's deoxygenated. That's it. Okay, so you have to use two different wavelengths and measure their intensity. And that's how you measure blood oxygen level. Yep.
Depending on the case, you also have to be careful with pulse oximetry because if someone were to have carbon monoxide poisoning, you can also present as 100% SpO2.
Okay. Did you hear that?
No, sir.
I'm sorry?
No, sir.
All right. Speak up a little louder.
Depending on the case, you also have to be careful with the SpO2 monitor because depending on if the patient has carbon monoxide poisoning, the SpO2 monitor can show higher numbers of blood oxygen concentration.
Okay. So, all right. And so that's how you measure oxygen levels, by measuring the intensity of red light and infrared light. How do you measure the pulse rate? Okay. So pulse rate is measured using the fact that your blood vessels expand every time your heart beats. There's more blood flowing through the blood vessel. There's more blood flowing through the blood vessel, and the absorption. So this intensity decreases up and down with time. Every time more blood flows, this intensity has decreased. Okay, irrespective of the level of oxygen. Okay, so that's how you measure the pulse rate, okay, just from the volume flow rate. Okay. So the oximeter detects changes in light absorption over time due to changes in volume of the blood flow. Okay, so here's how you define this. So this is oxygenated hemoglobin. This is the total hemoglobin, and if this is unoxygenated hemoglobin. Okay, so if 95%, if your SpO2 level is 95%, that means 95% of the hemoglobin are carrying oxygen molecules. Okay. So physics is a quantitative science, and we use mathematics all the time. Okay, and you saw an example of that. Remember, so if you wanted to control flow rate, if you wanted to increase flow rate, here was the formula for flow rate, and then you studied this formula to increase flow rate. What you realized was the best thing to do is increase the radius of the pipe. Okay. A small increase here, 10 to 15% increase in the radius of the pipe increases the flow rate tremendously. Okay, so after a big meal, where is all the blood gone? To your stomach. You don't want to be playing chess then, right? Because there's no blood flowing to the brain. So anyway, the body understands to adjust, it changes flow rate to different parts of the body when needed, as and when needed. Okay. So the laws of physics are stated as mathematical equations. Okay. So invention of much of modern biomedical technology depends on physics. Okay. So you saw how the scanning tunneling microscope works. So physics has been at the forefront of developing a wide variety of imaging technologies. So both biology is becoming increasingly quantitative. Mathematical models of enzyme kinematics provide quantitative predictions of complex biomedical paths. neural network models, okay, improve our understanding of both the real brains and artificial intelligence. Okay? So in science, they say, you guys, have you heard that the final frontier in science is what?
What was it?
What is the final frontier in science? Understanding your brain. Okay? Like I said, this is the most complex computer in the world. Okay? So now you want to use this to understand itself. Okay? So you have 90 billion neurons which are interconnected. Each neuron has roughly 10,000 connections. Okay? So neural networks model brain connection. So here's a model. So this could be a neuron. Here is the processing part. Here is the output, and so on. Okay. Epidemiological models increase our knowledge of how diseases spread, and they use mathematics to predict all of that. I mean, that's all math. Okay. Okay, so we might. Okay, I'll mention this one slide and we'll actually stop here. So diffusion is the process by which particles spread out from regions of higher concentration to regions of lower concentration, okay, due to random thermal motion. So diffusion is the most important process in biology. That is the process by which all molecules are moved short distances. Okay? Oxygen diffuses from your lungs into your blood, and neurotransmitters diffuse across synapses that connect. Okay, so yeah. So here is an alveoli, and when CO2 concentration is higher, it'll flow into alveoli. When oxygen concentration is higher there, it'll diffuse into the blood. Okay. And I'll give you numbers next time. Okay, so across these small distances, these molecules move. Here are the numbers. We'll talk about it again next time. They'll move across that distance in milliseconds. Okay, so that's how fast the molecules will move. So as long as you have small distances to move, they move in milliseconds. Okay. Diffusion is the process employed when molecules are transported small distances, roughly the size of a cell. Diffusion takes too long for transport across 10 centimeters. So 10 centimeters is about this big. Okay. So if oxygen had to diffuse across that, it would take 2 days. Okay. So whenever you're employing diffusion, it is to move stuff small distances. Soon as something becomes big, you have to have plumbing, okay, to transport stuff long distances so you can get close to the cells. Okay. All right, guys, we'll stop here today. So here's the deal. I'm going to finish lecturing on this chapter